In our previous video on cardiovascular physiology, we stated that the durations, ionic mechanisms, and the shape of action potentials varies in different parts of the heart. We were able to see the action potential in the ventricle of the heart. It is important you visit the previous video in order to have a good understanding. Don't forget to use the link on the description of this video. Now let's delve into what we have for today, which is action potential in the atria. The action potentials in the atria have the same features such as plateau phase and ionic mechanisms, just like that of the ventricles. You see why it is important you visit the previous video in order to have a better understanding. Aside the action potentials in the atria having the same features with the action potentials in the ventricles, the duration of action potentials in the atria is shorter when compared to that of the ventricles. In the atria, when all the phase 0 potentials are summed up, it is responsible for the P wave of the ECG. Phase 2 gives PR segment, and phase 3 corresponds to the atrial T wave of the repolarizations, which is not seen because it corresponds with the ventricular QRS complex, and it is also overshadowed by the ventricular QRS complex. Let's look at the automaticity of the heart. One of the unique things about the heart is that it has the ability to excite itself and does not depend on nerve stimulation in order to contract, unlike the skeletal muscles that requires nerve stimulations before it can eventually become excited. Visceral smooth muscles and certain neurons of reticular formation also have similar ability like the cardiac muscle. A freshly isolated heart or even slices of cardiac muscles can exhibit spontaneous contraction for some time. This inborn ability of the cardiac muscle to contract and relax on its own, that is the rhythmicity of the heart, is as a result of the presence of certain myocardial cells in well-defined areas of the heart, which can spontaneously depolarize, causing action potentials to be fired. These specialized areas are the SA node, the AV node, the bundle of His, and the Purkinje fibres. These specialized areas are collectively known as the special conducting tissues of the heart. Among these areas, the SA node depolarizes the fastest and then sets the pace for the heart, and for this reason, it is regarded as the pacemaker of the heart. Unlike the skeletal muscle, the heart cannot be tetanized. This is due to the refractory periods of the heart. During phase 0, 1, 2, and part of 3, before repolarization reaches threshold potential, the heart is absolutely refractory, and there is no stimulus, no matter how intense, can cause a response. Since systole ends in phase 3, the contractile response is over, while the heart is still absolutely refractory. This means that the heart cannot be tetanized like the skeletal muscle. This is functionally important because blood cannot be pumped by a heart in tetanic contraction. As the depolarization progresses beyond the threshold potential down to the end of phase 3, the heart enters a relative refractory period during which a strong stimulus can cause a response. Immediately following the relative refractory period is a period of supernormal excitability, when the heart is very vulnerable such that a relatively weak stimulus can evoke a response. Let us have a look at the variations of duration of action potentials with the heart rate. An increased heart rate brings about a decrease in the repolarization time of the heart, and this then shortens the duration of action potential. This means a shortening of the systole. Not only that this shortens the systole, it also shortens the rest period before the next cardiac beat, that is the diastolic period. Let us see the difference between the excitation-contraction coupling in the myocardium and the excitation-contraction coupling in the skeletal muscle. The excitation-contraction coupling is just the processes that link the depolarization of cell membrane to the contraction of the myofibrils. There is a slight variation of these processes in the myocardium when compared with that of the skeletal muscle. In the skeletal muscles, it involves the release of calcium ion from the sarcoplasmic reticulum, which then interacts with the troponin, thereby exposing the myosin binding site in the actin. But in the myocardium, the calcium ion is derived not only from the cisterns of the sarcoplasmic reticulum, but also from the extracellular fluid through the T-tubes, whose diameter is many times larger than that of the skeletal muscle. This extra supply of calcium is necessary for ensuring adequate contractile force of a muscle that works without stopping, like the myocardium. Earlier in this video, we stated that the inherent ability of the heart to contract and relax is due to the presence of pacemaker and special conducting tissues of the heart. Let us have a closer study of these tissues. Histologically, the cells of these special conducting tissues vary in size and character. In the sinoatrial and the atrioventricular nodes, there are small round cells which are the pacemaker cells, therefore are called the P cells. Other cells found in these tissues are often larger than the general contractile myocardiac cells. They have fewer myofibrils and mitochondria, a poorly developed sarcoplasmic reticulum, and high glycogen content. Now let's have a closer look at the sinoatrial node, that is the pacemaker. The sinoatrial node is located at the endocardial surface of the right atrium, near the junction of the superior vena cava and the right atrium. It contains P cells, transitional cells, and collagen fibers. P cells are responsible for the generation of cardiac impulse. The transitional cells are responsible for the conduction of the impulse generated by the P cells through the node, while the collagen fiber strengthens the SA node. The SA node has the highest rate of spontaneous depolarization, about 72 times per minute at rest. Therefore, impulse generated from it spread throughout atria and then both ventricles before any other tissue can originate impulse. It is because of this reason that the SA node is called the pacemaker of the heart. The heart rate is determined by it. The only time that the AV node or bundle of His can serve as the pacemaker of the heart is only when the SA node is destroyed. The SA node develops from the right side of the body and therefore receives its nerve supplies, which are the vagus and sympathetic. In the next video, guys, we will talk about other special conducting tissues like the atrioventricular node and the bundle of His. We will also have a look at the action potentials on these tissues, most especially the pacemaker tissues. Don't forget to subscribe, like, and share. We will meet in the next video.